Tonight, we have 3 of our incredible students from the course here to tell you about their graduate work. And their talks represent real research that's happening at the University of Washington right now. So we hope you enjoy this unique opportunity to get a behind-the-scenes look at it. So without further ado, I'll introduce our first speaker, who is Rory Mcguire. Rory does research at the intersection of computation and biology. Sometimes this means using DNA as a hard drive to store digital data, and sometimes this means using electronics to automate biological experiments. Rory has spent the last 2 years developing open source hardware and software with the aim of making biology and chemistry research more accessible, efficient, and equitable. So please welcome Rory.
Thanks all for showing up. Before I begin, I want to pose a question to the room. How many of you have ever tried to make your own pizza from scratch?
Not just tried.
Well, I've tried, emphasis on tried. And contrary to the impeccable rolling pin skills that I'm demonstrating in this photo, I'm not really much of a chef. So when I want a pizza these days, I typically opt for the easy route. I'm sure many of you are familiar with something like this. You select from a drop-down menu your desired ingredients or toppings. You navigate on over to the checkout button, and then usually in like 10 to 30 minutes, you have your very own bespoke pizza. It's fast, it's cheap, and you don't even need your own kitchen. But why am I talking about pizza other than the fact that it's like 5:30 and I've not had dinner yet? Well, I want to bring to scientific laboratory research the same convenience of scale, cost, and automation that I can get when I order a pizza online. How am I doing this? How am I going to do for laboratory research what the Domino's app has done for pizza consumption? Well, for some inspiration, I think we should have ourselves a bit of a history lesson. This is a photo taken in 1950 of a cell biology lab. It's pretty typical of what you would see in a research lab. It's a big room full of bottles and beakers and miscellaneous lab equipment. Lots of storage space, groups of scientists conducting experiments by hand, and of course all of this storage space and equipment and specialized labor costs a lot of money, so this sort of robust research could only really be done at very well-funded institutions. Jumping ahead a little bit, this is a picture taken in 1963 at Manchester University of the Atlas supercomputer. At the time, it was one of the world's most powerful computers, and I mean, it better be it's the size of a room. And it boasted a whopping 96 kilobytes of RAM and 576 kilobytes of storage. I hear some of you laughing, I'm being facetious. So just to give you some perspective of exactly how much that is, it would take over 35 room-sized Atlas computers to store the file of this PowerPoint, and to run this version of PowerPoint, it would take over 21,000 times as much memory. And of course, these days, we're all carrying around computers thousands of times more powerful than the Atlas just around in our pockets, and you can order pizza on the things, which is great. Now, let's see how far laboratory science has come in the past 70 years. Well, I mean, there's some new fancy lab equipment, yeah, but it kind of looks the same, right? It's a big room full of bottles and beakers and lots of storage space and lab benches where teams of scientists are still performing experiments by hand. And would you believe that just like back in the 1950s, this sort of robust scientific research is still relegated to the wealthiest of institutions? But what if we could shrink the lab? What if we could replicate the miniaturization of computers and put the lab onto a chip? Well, that may not make a whole lot of sense right now, but don't worry, we will get to the how of it all later. Right now, I want to talk about the why. What could be accomplished by shrinking down a lab onto a programmable chip. Some of my colleagues might rattle off a laundry list of potential benefits, but I believe that they can be boiled down to three primary incentives. First of these is the reduction of waste. Now, medical and research labs produce a lot of waste, usually in the form of what are known as disposables. These are often single-use plastics. You can think like face masks, surgical gloves, plastic test tubes, anything that gets used once and then is discarded to avoid contamination. A recent study at McGill University estimated that labs on their campus produce 100 tons of plastic waste every year. A similar study out of the University of Exeter estimated that labs just in their biosciences department produce 276 tons of plastic waste annually. And a commonly cited figure for globally produced laboratory plastic waste is 5.5 million metric tons of plastic waste every year. Now, for those of you who might not be as familiar with the metric system, to give you some perspective, that's roughly 61,000 blue whales worth of plastic waste every single year. And if we're talking about waste, then we should, of course, talk about biological waste or biohazardous waste. So any lab that works with biological specimens or samples like bacteria or blood is going to produce a significant amount of biological waste, even if they're not working with pathogenic strains or strains that cause disease. They're going to produce a lot of this kind of waste simply due to the fact that modern experiments require a significant amount of cell culture. And also because most of the time when something comes into contact with a biological sample, like a bacterial sample, and it needs to be discarded, it has to be considered as biological waste. And lastly, labs typically work with a lot of chemicals, usually in high concentrations. And these chemicals can be environmentally toxic if not disposed of correctly. But if we're able to shrink our experiments, then we would also be able to shrink the volumes of potentially harmful chemicals and biological samples that we need to use, which means that there would be less potentially hazardous waste, making scientific research safer both for the scientists doing the work and for the planet. The second incentive is lower cost. So of course, fewer required resources means lower cost per experiment. Even more cost savings can be achieved through automation. So if you can have an entire workflow run autonomously on a little chip, then there's not as much reason to pay for all of that workspace and storage space. And lastly, automation saves time. So normally science can be agonizingly slow. Chemical reactions can take hours to complete. Cell samples, if you're working with them, your cells can take days to grow. Experiments might have to be run dozens, maybe even hundreds of times to gather enough data or just to get your experiment to work correctly. And remember, most laboratory experimentation is still conducted by hand, but if the tedium of laboratory work can be automated, then that frees up the time of the scientists to pursue other things like analyzing their data or exploring new and interesting avenues of research. This could mean faster and less expensive drug development, or it could mean less downtime between your doctor's visits and getting your test results back. So a lab on a chip can really look like anything, as long as it shrinks down a laboratory workflow onto a single platform. A few non-exhaustive examples of devices that have come out of other labs in recent years include a UTI diagnostic kit that's built on a fidget spinner, or a chip with microchannels that simulates the functionality of a human organ, or a inexpensive diagnostic that can be printed on paper. Lab-on-a-chip devices are admittedly an emerging technology that still face a whole host of drawbacks and limitations, mostly stemming from the unique surface chemistry and physics that are at play at such a small scale. And a lot of lab-on-a-chip research these days is dedicated towards characterizing and overcoming these limitations. And it's but it is the potential advantages of these technologies that drives folks like myself to explore new and creative designs and solutions so that hopefully one day these devices might see everyday use. So the specific genre of labs that I'm interested in shrinking are molecular biology and molecular engineering labs. So these labs work with DNA and other tiny biological molecules to build something new and useful. Just to provide a few examples of work that you might see in some of these labs. Single-celled organisms can be turned into little factories by changing the DNA in their genetic code. With just the right combination of DNA, you can give bacteria the power to eat toxic waste, or you can make yeast cells that poop medicine. Or some labs might be interested in developing diagnostic kits out of molecules that will visibly change colors when they come into contact with viral components. And still others might be interested in using DNA as a way to store digital data in the same way that you might use a hard drive on a computer. So while I mentioned earlier that I'm not much of a chef, I will argue that the work of a molecular engineer or biologist is somewhat similar to that of a chef trying to come up with new dishes. So our scientist chef has an almost infinite variety of molecular ingredients to choose from, all of which may interact differently with one another. And with so many possible combinations of ingredients, the technology coming out of these fields is really only limited by the imagination and the financial and time constraints of those doing the work that often requires dozens or hundreds of rounds of experimental iteration. This makes these labs perfect candidates for chipification. So how are we actually doing this? You might have heard that DNA is the information molecule. It stores the instructions for making other molecules, like protein molecules, which are the workers of the cell that make sure everything runs smoothly. And these instructions are written in the rungs of the twisty ladder that is DNA, and they're written in a 4-letter alphabet. For our purposes, it doesn't really matter that it's 4 letters. But what matters is that each one of these rungs corresponds to one of those 4 letters, and the order, the sequence in which those letters appear, dictates the molecule that is being encoded for. So in this way, we can actually use strands of DNA to store experimental instructions, just like a recipe book, specifying which biological molecules are needed for a particular experiment. So now let's talk about, or let's say we wanted to create a mix of molecules that fluoresced if a sample from a COVID-positive patient is introduced. This end product, this specific mix of molecules, can be thought of as our molecular pizza. And the specific molecules can be thought of as the individual ingredients that we're adding to or topping our pizza with. And we'll keep the instructions for each ingredient in strands of DNA. If each strand of DNA holds the instructions for a unique molecular ingredient that we're adding to our pizza, then we obviously also need someone to read those instructions and follow them. Now, in a pizza parlor, this would be taken on by the cook. But in a molecular reaction, this is done by a special set of worker molecules called enzymes. In conventional laboratory workflows, the job of assembling and mixing these molecular ingredients often falls on the scientist. But if we provide these enzymes, these molecular workers, with all of the DNA strands that have the instructions for all of our desired ingredients, they'll do the job of generating and assembling all of the ingredients for us. So now, let's walk through an example of how a molecular engineering experiment might work in a typical lab today. In this experiment, let's say the scientist wants to find what specific combination of protein molecules can change sugar into fuel. The first step is to engineer some cells, usually bacteria, that are kept in large freezer units like this and engineer those cells to produce all of the desired proteins. The second step is to extract the proteins from those cells and then isolate them. And then experiments with all the different possible combinations of those proteins needs to be set up, run, and analyzed. And then usually experiments need to be optimized and repeated so that the scientists can get all of the levels just right. And again, keep in mind, most of the steps, most if not all of the steps that I just showed you, require manual mixing, measuring, and transferring of liquids. And in all, a single iteration of this process can take about a week to complete. In contrast, this is how the device that we're building in the lab that I work in is intended to function. So the device is going to look a little bit like a miniature golden checkerboard, with each square on the checkerboard being an individual electrode that can convey an electric charge. Attached to the surface of every electrode square are patches of DNA strands that hold the instructions for a unique molecular ingredient. Now, normally when these electrodes are in an off state, then the DNA sort of scrunches itself down, lays itself down flat against the surface of the electrode, becoming invisible to any of those enzyme worker molecules that might be floating around. So the side view that you're seeing here with the ovals, those ovals are the enzymes that we were talking about earlier that take the DNA and assemble all of the molecular ingredients. And right now they're just floating around in a thin layer of liquid on top of the electrode board, looking for DNA to read, but because all of the DNA is smushed down flat, they can't find any. But when our user decides what molecular ingredients they want made, they can make the selection on a computer just like one would make a selection of pizza toppings when ordering a pizza. And the charge of the corresponding electrodes will flip. The DNA on those electrodes will stand on end, hailing down any enzymes that are floating nearby. And then those enzymes will start reading the DNA, checking the order to see what ingredients need to be made. And then they'll start making intermediate molecules. And then finally make the desired molecular components or molecular ingredients. And then once all the ingredients have been assembled, the resulting output can be collected and analyzed. And the subsequent experiments, and any subsequent experiments can be set up to run automatically. So unlike the traditional workflow that we looked at right before this, only a single step, the step where the enzymes are loaded onto the chip, needs to be done manually. And this step takes only a few minutes. Everything else is done autonomously, and no large freezer units are needed to store cells or DNA. So we've talked about the advantages of this sort of lab-on-a-chip technology. We've talked about applications in molecular biology and engineering, and we've looked at how this sort of, or one of these devices might work. But to wrap up, I want to talk about who this technology is really for. So in 2016, I had the opportunity to study in Cuba at the University of Havana, where I worked alongside Cuban scientists and researchers and students in the biology department shown here. The folks I got to work with, the students and scientists and other researchers, were and are brilliant scientists doing invaluable work in their country. But lack of funding and difficulty getting supplies shipped internationally makes it extremely difficult to obtain resources which a lab in the US might take for granted. Even things like the disposable tips of pipettes, which are used for measuring and transferring liquids, are precious in Cuba. And experiments had to be planned in advance meticulously to ensure that as few of these pipette tips were used as possible. At a wealthier institution like UW, a researcher might not think twice about going through 100 of these tips in a single day. But in Cuba, this quantity of pipette tips might have to last a scientist 1 to 2 weeks. The embargo levied against Cuba by the US also makes obtaining internationally sourced chemicals and reagents extremely difficult. And work is further stifled by occasional rolling blackouts, making the stable storage of frozen samples difficult to impossible without a source of backup power. So lab-on-a-chip devices could potentially lessen the severity of these problems thanks to their small scale, reducing the quantity of chemicals and precious samples needed per experiment. Additionally, supplies necessary to build a device like our lab-on-a-chip tend to be more available than most lab supplies and reagents, which is good because anyone in the world is free to download and use our designs since we're keeping all of our hardware and software open source. Meaning that folks don't have to worry about copyright or patents, and again, all of our designs are freely available online. Keeping our designs open source is critical if we want our work to have the greatest positive impact. Lab-on-a-chip designs like ours also have a place in healthcare, especially in situations where mobility is critical. A field doctor attending to patients in rural areas without easy access to hospitals or other medical facilities might need on-demand access to a variety of pathogen tests to diagnose their patients, but transporting and storing dozens of tests that might need continual refrigeration isn't really feasible. So instead of carrying around a bunch of constantly expiring diagnostic tests wherever she goes, our field doctor can take the lab on a chip and use it to fabricate the components of whatever test she needs on demand and in single doses. So I'll go back to the question that I asked you at the beginning. What if we could shrink the lab from the size of a room to the size of your palm? Well, as it turns out, there's a number of answers to that one question. We can make science safer and more environmentally friendly through the reduction of waste. We can make science faster and simpler through automation. And we can make science more accessible and equitable by making our experiments less expensive and making our technology open source. With this technology, a future scientist might appear a lot closer to someone ordering a pizza online from home than someone in a lab coat at a lab bench mixing beakers of liquids together. By building automation into molecular research, we can make scientific discovery faster, less expensive, and more reliable for everyone. So I just want to thank everyone who I work with in my lab and all of the engaged folks for this wonderful opportunity to talk to you all.
All right, thanks Rory for a wonderful talk. Our second speaker is Keenan Ganz, who is a graduate student in remote sensing at the University of Washington. He uses specialized cameras on satellites and drones to study forest health and wildfire. One day, Keenan wants to build an improved forecasting system to understand when and where wildfires will burn next. So please welcome Keenan.
Okay, are we good there? We are. All right. Hi, everybody. My name's Keenan. I'm really excited to be here tonight because I get to talk to you about two of my favorite things, forests and fire. Now, in a second here, I'm going to show you a picture of a wildfire that burned in LA back in 2018. And when I show you this picture, I want you to focus on your immediate emotional reaction to it. Okay, ready? Here it is. If you're anything like me, that picture is both fascinating and terrifying. As people, we almost have a primal relationship with fire. When you see an image like that, it's almost like your ancestor from 10,000 years ago is reaching through time, grabbing your shoulder and saying, hey, you need to run from that. And this perception of fire as a destructive force isn't really helped all that much by what happens when the flames finally die out. Quiet, black, dead. We call these, well, at least the popular phrase for these kinds of post-fire forests is moonscapes. No life left. If this is how you view fire, as a destructive force that leaves nothing but death and destruction in its wake, then it's natural to fear fire, of course. But we got to this perception of fire through a very specific way. How many of you know Smokey the Bear? Smokey is perhaps the most successful public information campaign in history. Smokey has all sorts of iconic imagery telling us about wildfire. He tells us that care will prevent 9 out of 10 forest fires, that death rides the forest when people are careless. He says, why? Only you can prevent forest fires, and my personal favorite, our most shameful waste as he cradles Bambi. This kind of Smokey the Bear perception of wildfire can be distilled down to 3 ideas: that fire is avoidable, that fire is wasteful, and that fire is people's fault. And it's really worth dwelling on for a moment just how ingrained this is in our culture. I can conjure these ideas in your head with 2 words: only you. It really is amazing just how impressively Smokey has colored our perception of fire. But this Smokey the Bear approach to wildfire misses a lot of the complexity and the benefits that wildfire has in the Western United States. In fact, fire has been and will continue to be an important and natural part of the forests that we love here. But to understand that, we first have to understand how we got here. This perception of fire as destructive and evil starts with the perception that forests are a natural plantation. This idea came from Germany, which is the birthplace of modern forestry. And if you say that forests are a plantation, then, well, fire stops growth, it makes timber less valuable, and it destroys property. So if this is how you view the land, then fire is a complete evil and should be suppressed at all costs. For the last century, that's what we've done in this country. The United States developed an incredible infrastructure after World War II in the interest of protecting forests as a kind of national security. This was quite literally born out of the war effort during World War I and World War II. And think for a moment about all the resources you see in this picture. Personnel, roads going into the middle of nowhere, just in case we have to get out there to fight a fire. Helicopters, spotter towers built with helicopters on mountain peaks. It goes on and on. My brother, who lives in Idaho, was a part of this infrastructure. He was what's called a smoke jumper. He put on a 50-pound rucksack of the tools you need to fight fire in the middle of nowhere, got in the plane, and then parachuted into an actively burning forest to put it out. Suppress fire at all costs. But this kind of plantation forestry creates forests that look like this, all pretty much looking the same. And you can't quite tell from here, but if we got down on the ground there, we'd only see a couple of really valuable species, your Douglas firs, your western red cedars. But these aren't the kinds of forests that we want. This kind of plantation forestry lets forests grow for about 30 to 50 years before they are harvested. But in that time frame, the trees we have here are just getting started. This is the kind of forest that wants to grow in the Pacific Northwest. Well, I'm kind of lying there. This isn't quite the same species, but I'm using this picture for a different reason. Just to orient ourselves to scale here, that's a person down there. And yes, the trees are quite big in old growth forests. We love big trees. That's great. But don't focus necessarily on size here. Focus on the structural diversity. There's gaps in the canopy. There's all sorts of different kinds of trees at different ages. There's ferns, there's shrubs, there's broadleaf trees like maples, as well as needle trees like those Doug firs and cedars. These are forests that are both healthy, resilient to fire, and overall a much happier place to be than a constant green patchwork like you see here. So let's re-examine this Smokey the Bear approach to wildfire. First, is fire really people's fault? Well, Smokey's actually completely correct on this one. This is true. 80 to 90% of fires are caused by people, and that remaining 10 to 20% comes from processes like lightning, pretty much exclusively. But is fire wasteful? Absolutely not. Here's a fun science experiment you can do. If you're ever out in the woods, especially in a dry forest like we have in eastern Washington, and you find a pine cone that's like all sealed up like that, take it home and stick it in your microwave for 5 minutes. I'm serious. When that ends, you will be left with an extremely hot pine cone, but also hundreds and hundreds of seeds. This is called serotiny, and it's where a tree will invest tons of resources into putting hundreds of seeds in these cones and then sealing it tightly with resins and oils that don't let the cone open up until the cone gets to several hundred degrees Fahrenheit. That only happens in fire. And these banks of seeds that live in the forest canopy are just waiting for the next time the forest burns to get dropped onto the ground and start the next generation. Fire is part of the circle of life. For humans as well, fire is a tool. Indigenous cultures in the western United States, up and down the Pacific coast, have used fire to their advantage for thousands of years. This is to both create habitat for helpful plants like huckleberry, or to create habitat for game animals, as well as supporting the overall health of the forest in the long term. Now, last thing we'll talk about here for is fire wasteful, let's go back to this moonscape picture. Imagine what this forest looked like before it burned and compare it to what you see now. What percent of the woody material do you think is still there after the fire has burned? Half? 3/4? It turns out the answer is over 90% of woody material remains after a fire. And this was a really intense one even. In some fires, that number can be as high as 99%. So all that carbon isn't getting emitted into the air as greenhouse gas emissions. It's staying in the form of these dead trees that we call snags. These create habitat for small mammals like squirrels and birds, as well as insects. And in 100 years, when those snags finally fall over, all that carbon and nutrients will just get absorbed into the soil, fertilizing and supporting the forest as it's growing. You can even see the start of that new forest if you look closely at the bottom there in those wisps of green. So fire is not wasteful. But now here comes the hardest question of all. Is fire avoidable? Fire burns our houses down, it can hurt people. Do we have to have it? And the answer, my friends, is no. There is additional complexity to this, and I want to preface that before I give you a blunt yes or no response. But in general, our policy of suppressing fire for the past century has only engendered larger, more dangerous, and more destructive fires today. But again, go back to this picture. This is terrifying. There are probably people whose homes are nearby to this place, or others who narrowly escaped the mountains before the flames caught up to them. There's probably someone in this room who's been negatively affected by fire in some way. And this is the push and pull that we face now in managing our modern forests, is fire is a part of these ecosystems, but we as people want to avoid fire at all costs. So what do we do? Well, to know what we do about fire today, we first have to understand, how does fire work in Washington? And this, my friends, is a tale of two different kinds of forests. In Washington specifically, our state is neatly cut in half by the Cascade Mountains, and that has an interesting effect on the kind of weather that we see. The predominant wind pattern here is to blow moist marine air from the Pacific into the Cascades. Now, those clouds will hit the Cascades, and, well, the only way they can go is up. And as those clouds rise, they can't hold on to water quite as well. So it all comes out as the rain that we all know and love, and it creates incredibly dense, lush, beautiful forests that give us the name, the Emerald City. But by the time we get to the eastern side of the state, those clouds have pretty much lost all their water. And so the forest that forms out there is very sparse. There's not as many trees, and it's very, very dry. Now you can imagine that these two kinds of climate patterns create different kinds of fire. Let's start on the west side. To have fire here on the west side, it's really quite rare. Forests here burn generally once a century, and if you go out onto the Olympic Peninsula or other really, really wet areas, you can go over 1,000 years before fire burns. You need three things for fire to happen: dry, hot, late summer weather, easterly winds, so winds blowing from east to west, and then an ignition source like lightning or like a silly person with a firework. 1 and 3 pretty much happen all the time, but 2 is really rare. Because remember, I just told you, the wind goes the other way. So that's why fire is so rare in this part of the state, is you need the wind to flip directions and also have incredibly dry, very hot weather. And this is so rare that we often liken fire here on the west side to rolling the dice. Every year, it's not very likely you'll get fire, but eventually you'll roll snake eyes. And when that happens, it burns. Because the forests here burn so rarely that it's not really worth adapting to try to survive fire. And we call these high severity fires because they're generally killing lots of the trees. The flames are all up in the canopy as well. But on the east side, the story is much different. Because it's so dry out there, they burn much, much more often, typically every 10 to 25 years. But they don't really kill as many trees, and there's a couple of reasons why. This is a prescribed burn, so this fire was set by people, but its behavior is really similar to natural fires. I want to draw your attention to some of those trees in the back. The kinds of trees you see on the east side of the state, so your ponderosa pines, especially are really common, have incredibly thick bark, like 2 to 3 inches thick. And that provides insulation that prevents the living part of the tree on the inside from being killed by the flames. But also notice, there's no branches close to the ground. That wasn't done by people. The trees do that themselves to reduce the likelihood that flames will catch on a lower part of the canopy and climb up into the crown, where they can actually kill the tree. So fires like this, most of those established trees don't really care. They can survive it just fine. But in these fire patterns, we also have climate change. In general, climate change rears its ugly head and it makes our state warmer and drier. Here on the west side, that means that we're effectively loading the dice to make those already rare fire conditions just a little bit more frequent. And on the east side, climate change means that forests are recovering more slowly after fire, or not recovering at all and turning into grasses. And large, destructive fires are becoming more likely. But is this normal? Is the kind of horrible, smoky, ashy weather we seem to be dealing with all the time in August, is this the apocalypse, or is this part of living here? And it turns out that, yes, this is normal for this part of the state. We have evidence of large fires that would have created incredibly smoky conditions in what would become Seattle happening hundreds of years ago, long before European settlement and our modern approach to forestry created larger, more destructive fires. But this kind of weather and smoke is happening more often, both because of climate change and because of our fire suppression history. So I've given you the story so far, and the question now is, how can we live with fire in Washington? And that comes back to the kind of diversity we see in old-growth, unmanaged forests in this part of the state. This isn't true so much on the west side, generally because, like I said, the forests here grow to be very dense and lush, so when they burn, they're going to burn like crazy pretty much no matter what. But on the west side, having this on the east side, having more of this structural diversity, gaps in the canopy, lots of space between the trees, instead of growing those forests incredibly dense and thick, makes them more likely to survive after a fire and less likely to be incredibly destructive and smoky, which is what we don't want. And the new kind of forestry that is supporting this kind of forest development is called ecological forestry. And this approach creates structural diversity while still making money off of forests. The goal here is to emulate the natural processes that create diversity in forests. This takes the form of leaving behind clumps of trees, either in discrete little patches, like in the top of that picture, or in dispersed kind of salt and pepper pattern, like in the bottom of that picture. It means leaving trees behind around streams and other riparian corridors so that we don't erode away all our topsoil. And it means, yes, intentionally setting the forest on fire, because this kind of low severity fire that trees will survive is how these forests were meant to be. So my research in particular asks, if we have to live with wildfire, can we predict where fires will burn? And my approach to this question is, if drought is needed to ignite wildfire, can we measure drought to predict fire? But what exactly do I mean by measuring drought? To understand that, we have to take a little look into how trees make their living. So under regular conditions, so a tree or really any plant with plenty of water, we're going to zoom in on the leaf. And top of that diagram is the outside of the leaf, bottom of the diagram is the inside. And sort of on that border space, you'll notice there's little holes in the leaf. And those are called stomata, and they allow gases to move between the inside and the outside. We have 4 characters in this story: carbon in the form of carbon dioxide gas, oxygen in the form of oxygen gas, water in the form of, water, and sugar. What happens under normal conditions is that carbon will enter the leaf, combine with water to make oxygen and sugar. The plant will keep the sugar, that's its food, and the oxygen gets released out into the air for us to breathe. It's a pretty sweet system. But what you may not know about this process, which is photosynthesis, is that for every turn of this crank, for every one sugar you make, you're losing hundreds and hundreds of water molecules. Because again, those are open holes in the leaf. Water is free to evaporate out. And it turns out that there is so much of water just lost to evaporation that for a Douglas fir tree in the middle of summer to make 4 sugar cubes worth of sugar, it has to use about a gallon of water. And the vast majority of that water is not going into the sugar. It's just evaporating into the air, getting lost. Now, when drought happens, the leaf closes. The plant has to keep as much water as possible to stay alive, and so it stops making sugar, but also holds onto its water. And it turns out that different rates of evaporation of water affects the temperature of plants. To understand why, think about some time you've been swimming in the middle of summer. Even if it's like 90 degrees outside, when you first come out of the water, you're cold. And that's because water is evaporating off of your skin and sucking heat away from you. It turns out the same thing happens in forest canopies. Trees that are happily drinking water and making sugar like there's no tomorrow are much cooler than trees that are struggling with drought and closing their leaves. So my question is, are hotter forests more likely to burn in the next couple of months or couple of years? And I hate to leave you on a cliffhanger, but I don't know. PhDs take a long time to complete, and I want to give you a more complete answer to that question in a couple of years. But if there's nothing else I want you to learn from this presentation, it's that fire is a part of living in the Pacific Northwest. And every forest you see here started as one that had just burned down. And every forest you see here will burn again one day. It's not a matter of if, it's a matter of when and where. So next time you see a huge wildfire in the news, it's not the apocalypse. It is a natural part of our forests that we need to be ready to respond to protect human health, but also preserve our environment. Thank you very much for listening.
Whoa, that's really tall now. Yeah, I don't know. You might have to fix it again. I'm not sure. Thank you, Keenan, for a great talk. Our last but definitely not least speaker of the night is Rasika Venkataraman, who's a graduate student in the Department of Laboratory Medicine and Pathology. Her research focuses on studying a specific hereditary mutation in our DNA that causes blood cancers. And she aims to investigate how this mutation alters the environment in which cancer cells develop and grow to improve our ability to treat these blood cancers. So please welcome Rasika.
Thank you all for being here. And I'm going to talk to you about mutations and cancer and how investigating the environment in which the cancer grows could be beneficial for treatment. So to get started, I want you all to take a few seconds to think of your favorite comic book superheroes. And if you're anything like me, what might have come to mind are the X-Men or the Hulk. And while the X-Men and the Hulk have these fancy, cool superpowers, the way they got these powers is quite different. In the case of the X-Men, they were born with a mutation in their X gene, which gave them their superpowers. And with the Hulk, Bruce Banner acquired these superpowers at some point in his life when he was exposed to gamma radiations. This, of course, is in the fantasy world where when you're born with or you acquire a mutation at some point in your life, they give you cool superpowers and people make movies about you. But in the real world, this is not really the case. In the real world, people are born with some set of mutations which are called inherited mutations. And as the name suggests, these are typically present at birth, and we inherit these mutations from our parents. On the other hand, acquired mutations, typically we obtain with age as we grow older. And these mutations come from either random spontaneous occurrences or by exposure to a lot of environmental factors such as smoking or exposure to certain chemicals. And unlike the fantasy world where the mutations give you superpowers, in the real world, mutations either have no effect on our health, sometimes they can be mildly beneficial, and sometimes they can cause harm, causing diseases for which we may or may not have cures. But before we talk more about inherited and acquired mutations, let's talk about what is a mutation. So the instructions to how our body functions is basically stored in these codes called genes, which make up the DNA, which is unique to every one of us. And just like how you need a chef or a cook to kind of read the recipes in a recipe book, you can think of genes as the different recipes in a recipe book, and the genome is basically a collection of genes, much like how the recipe book is a collection of recipes. And the recipes can be written in different languages, for example, English, and so the language in which this information is stored in our genes is written in the language of DNA. So like you need a chef to read the different recipes in a recipe book, you have this intermediate molecule called the RNA, which Rory spoke about, and you need this to make the final dish, if you will. Like the chef can make different dishes from different recipes, which can have different purposes, like a main course, a refreshing beverage, or even a dessert. The information in the genes leads to the formation of this final dish, if you will, which is called the protein, which has many different functions in our cells and bodies. You can think of a protein as a worker in a factory who's doing a whole bunch of different things. In the case of a mutation is basically an error or change in our DNA, much like how you can have an error in your recipe. You could have a typo, or the pages may be stuck together or torn in your recipe book, which basically makes the chef read the recipe in a wrong way, and the final dish that you create turns out bad. For example, if the typo said add salt instead of sugar while making an apple pie, you may end up with a really salty pie, and that's no good. Just like that, when you have a mutated or erroneous DNA, it can lead to a mutated protein, which changes the function of the protein. So what do I really mean by change in protein function? So proteins basically do everything in our body. So this is our DNA, which we spoke about. It leads to the formation of a protein. And a protein has many different functions in this microscopic unit of life called the cell. And a group of cells together make different organs in our body, such as the liver, the kidney, our bones, which make us who we are as human beings. So when you have a mutation in your DNA, you have the mutation carried forward in the protein, which then ends up affecting or changing its functions across the different organs in our body. And like I said before, this mutation could be mildly beneficial, it could be neutral, and sometimes it does cause harm and lead to diseases such as cancer. So what exactly is cancer? So when you have normal cells in your body, when they sometimes acquire or inherit these mutations, they basically end up dividing much faster and aggressively at the cost of normal cells, and they just take over, and this is what a cancer cell is. And this then leads to cancer in different organs or parts of our body, which all of us are quite familiar with. So going back to these inherited and acquired mutations, the inherited mutations are basically present in all cells and organs of our body. You can think of this like the typo in the recipe book. So for example, if the typo was present in the original edition of the book, it's going to get passed on to all the multiple reprints or photocopies of that book. Just like that, the inherited mutation is present in all cells and organs in our body, and we can pass it on through generations. In the case of acquired mutations, these mutations are specific to an organ. So for example, it can either occur only in the kidney or only in the liver, much like how when you have the pages stuck together in a recipe book, that's pretty unique to that one recipe book that you're holding. It's not going to be passed on to the next photocopy or reprint of the recipe book. And when mutations do cause harm, they can cause different diseases. And the examples of some diseases which are caused by inherited mutations are cystic fibrosis, sickle cell disease, and also a risk of cancer. On the other hand, acquired mutations typically cause different cancers in different organs of our body. So you may notice here that cancer is caused by both inherited and acquired mutations. And one such cancer that is caused by both these types of mutations is blood cancer, and specifically acute myeloid leukemia, or AML, which is one type of blood cancer and which is what I'm interested in studying as part of my graduate work. So if any of you were here a couple of weeks ago, you would have heard Elizabeth talk about blood cell formation and how this is a very intricate and balanced process in our body. So in the case of blood cancer, this balance is basically thrown out of whack. And when a patient has blood cancer, their blood has a decreased ability to carry oxygen to different parts of the body, which makes them feel tired or fatigued. The blood also doesn't clot as well, so even if we have a small prick or a cut, it leads to excessive bleeding, which can be really dangerous. And also the patient has an increased susceptibility to infections because now your blood cells are not really great at protecting you from viruses and bacteria. And of course, there is an increase in cancer blood cells at the cost of regular blood cells in your body. So in the US, there are almost 20,000 new cases of AML or blood cancer that are being diagnosed every year. And about 5% of these AML are caused by an inherited mutation in the gene DDX41. So DDX41 is the most common inherited mutation that puts us at risk to developing blood cancer, or AML, as shown in this graph here. Just below DDX41 is also a series of other genes which, when mutated with inheritance, leads to AML, but as you can see, DDX41 is the most predominant. And also the blood cancers that are caused because of the DDX41 mutation is typically seen in older patients when they're about 60 to 70 years of age. So now if we look at a person who is born with a healthy DDX41 DNA, So all of us actually have 2 copies of each gene in our body. So if you take any gene, for example here DDX41, we have 1 copy of the gene from the mother and 1 copy of the gene from the father. And this is in case of a person who has no mutation. So when a person is born with an inherited DDX41 mutation, 1 copy of their gene is mutated. And this copy could be either from the mother or the father. And typically, these people who have this inherited mutation don't really show any disease symptoms, and they lead a healthy life for most of their life. But as they approach that 60, 70 years of age, what happens is they get a second mutation in the second allele, second copy of the gene of DDX41, which basically makes the DDX41 protein mutated, and this leads to blood cancer. So you might be wondering, okay, so what is the function of DDX41 protein? Because you said before that all proteins have different functions in our body. So this is still a bit unclear to us, but one of the main functions of DDX41 is to respond to viral infections and viruses in our body. And other than that, it basically plays a role in basic maintenance of cell functions, or like as a housekeeping, it plays a role in housekeeping the cell, which is not really great because now it has so many different functions, and it's hard to really pinpoint what is the function that's going bad when you have a mutation which is causing cancer. And that's what we're all trying to study. And like I said, we still quite don't understand why when a patient has an inherited mutation, they end up acquiring that second mutation when they grow old. And we also quite don't understand why, when you have that second mutation, it's leading to blood cancer. So the current treatment for DDX41 blood cancer is like treating any other type of blood cancer. So typically, the patient shows up in the clinic with the symptoms that we spoke about earlier. The doctor then runs a diagnosis and then detects that the patient has a DDX41 mutation and diagnoses that the patient has the symptoms for blood cancer. The patient then undergoes different treatment choices and therapy cycles, which are typically radiation and high-dose chemotherapy to get rid of these bad cancerous cells, followed by something called a blood transfusion, where the cancerous blood cells are removed from the patient and replaced with healthy cells from a healthy donor. And a lot of times, there's also an extreme form of treatment that is required called the bone marrow transplant, where basically blood-forming cells are directly removed from the patient's bones and replaced with healthy blood cells to try and cure the patient. We'll talk in a minute about what a bone marrow is. So this seems really complicated, and definitely there are a lot of problems with the current therapy for blood cancer. It's really invasive, especially in the bone marrow transplant that I spoke about. It's very, very expensive. And also, not everyone is a match or eligible for transplant a lot of times because of lack of donors or mismatch between donors. And additionally, since these patients are typically of older age, they also have a lot of age-related health conditions such as heart disease or autoimmune diseases, which just makes them difficult to undergo this kind of treatment. And even if they do undergo treatment, within a few years, the cancer returns in about 50 to 60% of the patients. This is not really great, is it? So scientists like me and doctors in the field of blood cancer are using research to try and really fix this problem, or try and come up with at least better treatments that we can use either along with these treatments or independently to improve the current treatment. And so as part of my graduate work, I'm trying to look at how we can study these blood cancer cells to treat them, not by just looking at them, but maybe looking at things around them. To do that, I'm trying to look at the environment in which these blood cells and cancer cells are growing. So blood cells develop and grow, grow and mature in a spongy environment called the bone marrow, which is present within our bones. So the blood cells are really nurtured by the environment of this bone marrow, much like how plants are nurtured by the environment of the soil. And I think we can all agree that healthy soil should equate healthy plant. If we zoom into the bone marrow environment, which is shown on the right there, the environment is actually quite complex. Just like how the soil environment is made up of different factors that affects its health, such as water, nutrients, pests, bacteria, and fungi, either good or bad, the bone marrow environment also has multiple factors that play a role in its health. So as you see here, the bone marrow environment has bone cells, fat cells, structural cells, and all of these cells are in constant communication with each other, and by releasing this growth signal or environmental cues that are really crucial for blood cell formation and growth and development. So you can think of these growth signals as environmental cues for the blood, just how components in the soil can be environmental cues for the growth of the plant. So now if we look at a healthy bone marrow with no DDX41 mutation, these are again all those components in the environment that I spoke about. The environmental cues or growth signals that are released from these healthy cells lead to healthy blood cell formation, much like how a healthy plant will grow in a healthy soil. So I do want to preface by saying that we really don't know what's happening in a DDX41 mutated bone marrow environment, and that's kind of what I'm trying to study as part of my PhD. And my research is kind of trying to understand if we can learn more about the mutated bone marrow environment and if that'll help us improve treatment. So one idea that I think may be at play is that in the case of a DDX41 mutated bone marrow, so remember that the inherited mutation is basically present in all the cells in our body. So all of these bone marrow environment cells will all have the mutation. So my hypothesis, which is basically like an educated guess or claim from past scientific data, is that these mutated cells, the growth factors and environmental cues that they release basically end up being corrupted, and they could possibly corrupt the blood cells that are growing in the environment, leading to blood cancer. Much like how when you have a defective or bad soil, after a point of time, the plant may also get diseased. So the question arises, are defective signals or cues in the bone marrow microenvironment, when mutated, are these corrupting blood cell development leading to cancer? So in the lab, what I'm trying to do is basically study these bone marrow cells and ask the question, are the DDX41 mutated bone marrow cells any different? So I grow these bone marrow cells in small Petri dishes in the lab. These dishes are made of plastic, and you add the required food or nutrients for the cells to grow, and they grow really well. And I have two sets of these cells, one which don't have any mutation, and I call that the healthy set, and one which has the DDX41 mutation. So what I'm trying to ask is, can I identify corrupt growth signals in the DDX41 mutated bone marrow cells? And what I actually found was indeed true, indeed the same thing, that these DDX41 mutated bone marrow cells actually have an increased presence of these corrupt growth signals in them, which is shown by the orange bar in the graph there, as compared to the healthy cells, which have either lesser or almost negligible corrupt growth signals, which is shown by the black bar there. So the idea is that if we are able to identify these corrupt growth signals in the mutated bone marrow, can we use drugs to basically try and block them or reduce their presence to kind of fix the defective bone marrow environment? So next, what I can also do is kind of throw blood cells into this mix. Now I can grow the blood cells and bone marrow cells together in the lab and to kind of mimic the bone marrow environment. And then I can kind of use these drugs to add them to these cells and really ask the question, how is the blood cell developing now once we add these drugs? If blocking these signals will improve the blood cell health. There are also multiple other ways in which you can study the bone marrow environment by looking at all these other complex cells that I spoke about earlier, the fat cells or the bone cells. And currently using a lot of advanced techniques, you can kind of ID these different kind of cells, pull them together, and really ask the question, who is or which type of cell is making the bone marrow environment go bad. So going back to the clinic and treatment of blood cancer, so the typical treatment, as I mentioned, is a blood transfusion or a bone marrow transplant, where these diseased cancerous cells are removed from the patient's body and replaced with healthy cells from a healthy person. Much like how you can treat a diseased plant by replacing it with a healthy plant. And this may really kind of help the healthy plant grow. But also, as we spoke about earlier, the cancer does return in a lot of these patients who undergo this treatment. Much like how when you have a defective or faulty soil, your plant has a lot of possibility to also get diseased over time. So the question arises, can we treat the defective bone marrow environment? Much like how you can treat the soil with pesticides when there are pests, or if there's a lack of nutrients in your soil, you can add fertilizers to improve its nutrient content, which will then enable the new plant to also grow and thrive. So what I'm trying to ask is, can I use these drugs to really treat the bone marrow environment so that they end up blocking these bad, corrupt growth signals, and thus, if we can prevent these blood cells from becoming cancerous? So where does my research fit in the broad picture of blood cancer treatment, especially in the case of DDX41 mutation? So this is the figure that we saw earlier, where the patient appears with the symptoms to the doctor, they undergo a diagnosis, and then they undergo a lot of treatment and therapy cycles. And the goal of my research is to really fit in here, where we could try and use drugs to kind of correct the faulty or defective bone marrow environment, either by itself or in combination with the previously existing treatment, with the ultimate goal of really increasing survivorship of these cancer patients and decreasing disease progression. Thank you all for listening. And I really want to thank the Stanley Lee Lab at Fred Hutch. A lot of people at the Lee Lab are studying the different mutations that cause blood cancer and trying to come up with better ways that we can improve treatment. And I also like to thank Engage Science for this really cool class this past quarter. And a big shout out to Nicole, who's been like an amazing instructor and helped me get over my stage fear. Thanks.
All right, thanks, Rasika, for an awesome talk. We're going to transition to our Q&A. So please take a minute to just relax and think about all of the questions that you want to ask our awesome speakers while we rearrange the stage. Okay, I'm supposed to be moderating this Q&A session, but also I have to fix the camera in the back, so sorry for the weirdness. We have a mic over here on the side for you to come up and ask questions. I know nobody wants to come and talk into a microphone, but we would love to have the audio of all of this, so please don't be shy. You're not in the camera shot, so you'll just hear a disembodied voice in whatever happens later on. And we'd love to take any questions that you have for any of our speakers. Don't go all at once.
Hi there. Can you hear me? Yeah, I think the first speaker and the third speaker, you guys showed the same analogy, but there was a small difference. I think the first person, you referred to the chef as the enzyme. The third person said it was, the third speaker said it was the mRNA. So are they the same, or they're different, or there's enough?
Can you hear me?
Yeah.
Okay, so the mRNA or the enzyme are basically intermediates, which all play a role at that intermediate level between the gene and the protein. So I use the analogy of the chef with the mRNA, but you could think of multiple different chefs or multiple different cooks. One is the mRNA, one is the enzyme, if that makes sense.
So they do the same thing?
So they all work together to be able to make the final protein. So I'm trying to think what would be the better. So you could think of it like a assembly line in maybe a restaurant where you have the main chef and some people who prepare the basic ingredients and some people who do the next step of the dish. So the mRNA could be the first person who's cutting the ingredients, and the enzyme could be the next person who's adding on to that, if that makes sense. But at the end of the day, all of these intermediates are working together to make the protein. And I think I just simplified it as the mRNA, and Rory did with the enzyme. Okay. Do you have anything to add?
I was just going to add that the mRNA is sometimes analogized as like a photocopy of the DNA. But I think it also can make sense, depending on what you're talking about, to think of it as like a cook in a kitchen, because it's the molecule that relays the information from sort of the data center that is the DNA to the molecules that actually generate the proteins, which is the end product. So it's in that way, it's carrying out the actual relaying of the information.
Yeah.
So for the talk about if hotter forests are more likely to burn, sort of a question that comes up is like, what do you mean by like a hotter forest? Do you have like one place in that forest where you're measuring temperature, or how might you like look at the temperature of that entire forest?
Yeah, that's a really good question. Fortunately, we have NASA, which means that we can put cameras in space that actually do that imaging for us. And it's kind of a tricky task there, because those satellites orbit at different rates, and sort of the size of things that you can see in each of their images differs. Or like in some, you could see like maybe individual buildings of Seattle from space, but in others, you might have one neighborhood all blended together into one big blob. So we combine some of those like larger scale images from satellites with, really small measurements, like even down to a single point, like we put a thermometer in, or we can fly a drone with a thermal camera on it and create like a slightly larger area. It all comes down to what question we're trying to answer and what data is best suited to answering that question.
Thanks.
And mine is a follow-up to that exact same question. What kind of lead time are you talking about? Is it, when you look at your data, do you go, holy moly, evacuate that neighborhood? Or are you looking at it past tense, going, oh yeah, that's why that one burned? What's your time frame?
Sure. So to answer that question, I should mention that sort of the state-of-the-art fire monitoring system that we have in the United States, it generally forecasts like a week to like a month at best. And, at a month, it's like, we're not looking at like the Mount Baker Snoqualmie forest, right? It's all of Washington is like one data point looking a month out. We have a daily forecasting system. If you've ever seen Smokey the Bear pointing at like a sign, that's like a national like system. And it forecasts about 24 hours into the future. But the thing is, that's good for people to know so they don't set off fireworks or gender reveal parties or anything. But if you're someone who's managing that forest and you have to like hire people or get a helicopter ready to fight that fire, you probably want to know about that a little bit more than 24 hours to a month in advance. So I'm targeting 6 months to a year as the lead time, because that would give people plenty of time to say, all right, Northern Sierra Nevada, that's probably going to be the problem area this year. So let's allocate more resources there.
So this weekend's warm weather wouldn't trigger your...
It might.
I actually also have a question about the fire suppression. So you mentioned that forestry has changed from like plantation forestry to this more ecological system. Do you feel like information about forestry has also changed, right? Like Smokey the Bear was so pervasive, and do you feel like there are there's new information making it out to people that forestry has changed?
Yeah, so that change really kind of started in the 80s. If any of you were around when the controversy over the spotted owl started, that was kind of the beginning. And that push was not necessarily the spotted owl is extremely important and we need to conserve it. It was the spotted owl is an instrument with which we can start to implement these ecological practices, because that's just how the laws were written, right? There wasn't a law that said, we need to create structural diversity in forests. The law said, we want to conserve endangered species. And the owl was a way to do that. That then grew into the Northwest Forest Plan, which is sort of a, Washington, Oregon, Northern California wide plan about how to manage forests. And I think that those very public legal battles definitely brought the issue to the forefront. As far as, was that a well-reasoned, nuanced discussion of the issues at hand? Perhaps not. But one thing I really love about the Pacific Northwest is that we care about this natural heritage. And all of you are coming to talks like this and you're learning the nitty-gritty details. So yes, I think it is improving.
So I have a question for Mr. Chip.
Yes.
I'm fascinated by that. But if by the time you get your DNA, it goes back to your chemistry and physics of getting your DNA on your chip. So after you've GM'd it on there, haven't you changed it enough that what you're getting for answers is not replicating what you would get in the lab doing it the old-fashioned way? That's your question. And then Miss C over there with the transplant. Do people relapse even with a bone marrow transplant in 3 years? I mean, I would think a transplant would have fixed the issue, and that would have helped it.
Yeah.
I can't think of a fire.
Excellent questions. The DNA that we're dealing with is it's fairly long. DNA, if you remember, I was talking about is the data is written in rungs. Those rungs are often referred to as bases. So the DNA that I'm working with can be thousands of bases long, and that's basically so that every single strand is one gene, so it produces one protein. And the way DNA works is that there's always a start of the DNA. That's where it starts getting read. Like if you're thinking about it like a page of a book, that would be the top left. That's where you start reading. And if we orient the DNA so that that starting point is way up near the top and not near the bottom, then the modifications that we're making to the DNA to jam it on aren't really, they don't impact the protein that's being created.
Thank you for your question. Yes, even after the bone marrow transplant, you do have relapse, typically 3 or 4 years or even after that, which is basically the argument for if the soil is corrupt, your plant is probably also going to get corrupt over time. But again, it needn't only be the bone marrow environment that could be defective. A lot of times, even from the donor cells, if they have some mutations in them which were not caught earlier, could develop into blood cancer. Because then you call that donor-derived blood cancer. But it's hard to really distinguish where the cancer is coming from a lot of times when there is a relapse, but there is a relapse even after the transplant.
Thank you very much, all of you. It was very interesting. Rasika, I have a couple of questions about that corrupt growth factor. One is, are they other than bone cancers? Is the corrupt bone factor present there, even though it's obviously not coming from the bone marrow? Or does it vary within the bone marrow?
So is your question that are these corrupt growth factors also present in other parts of the body?
In other types of cancers, other than bone cancer?
Yes.
Or blood cancer, I mean.
Yes, they are. So their overall called inflammation-based signals, which basically is when your body and cells are not happy, and that's the response that they produce. So it is not specific to the bone marrow environment, which is why we would have drugs that are already present to kind of block them in other types of cancers or autoimmune diseases, if you will. And we can leverage that to try and use the same thing in the context of a blood cancer when the bone marrow environment is defective.
Great. And let's see, my other question was also concerning that. So is it possible that the corrupt growth factors are influenced by environmental causes? In other words, like enhanced in such a way that if someone were exposed to air pollution or really poor quality food or that kind of thing, that then that inherited mutation would be more likely to be, develop in the other chromosome?
That's a wonderful question, and we don't know the answer to that. Because basically we don't know why this second mutation arises. We just know that it arises randomly at some point in life and gives you cancer. And we still don't know what are those factors that are driving it to get that second mutation. So we're working on that.
Great, wonderful, thank you.
We've got time for one more question.
Did you consider any other types of experiments for the lab on the chip? You mentioned that you thought molecular biology and engineering were particularly well suited, but were there any ideas that you discarded?
Not discarded. I mean, it's just that the lab that I work in and the folks that I work with are particularly interested in that sort of thing. But the variety of things you can do with lab on a chip are pretty astonishing. Like I mentioned earlier, that there's this whole subgenre of labs on a chip that are called organs on a chip, and they're built so that the flow of liquid through these little chips, they pass over cell growths in a way that replicates the functions of organs, so that you can, rather than testing a drug on mice or something, or just testing it on some cells in a petri dish, you can run your drugs through this system, and it'll give you a more accurate outcome of what the drug is doing to that organ without actually having to test it on a live animal. So there's so many things you can do with it. It's just we had to settle on one. So.
Awesome. So thank you all again for joining us tonight. We would love to invite you back for our final Engage Science Now event, which is on May 22nd, next Monday, starting at 5:30 back here. If you want to learn more about Engage, please feel free to come chat with me after this. You can also visit our website, which is engage-science.space, or you can follow at Engage Science on Instagram or Twitter. If you're a graduate student and you're interested in learning more about science communication, this course will be offered again next winter, and applications typically open in November, so look out for those. And if you have more questions for our speakers tonight, you might be able to catch them after this for a few minutes if I'm not volunteering too much from them. Thank you all for coming.